because I can advance it just with my arrow key.
Okay. And then you can advance with arrow here, or the mouse, or this would advance it too.
Key as well. So that's where I've got the corresponding slide that should be on the screen, so it's visible as well.
Okay. All right.
So
And since we have 30 minutes of discussion after your talk, if it takes 30 seconds or a minute for you to get set up, we're under no real time crunch. So don't feel pressured. I'll help you get set up so we get you started.
all right, if I leave this up here then?
Yep, for sure.
Leave that, and I'll take
And
everything.
I should ask you, how do you pronounce your last name? As someone who has an unusual last name, I want to try and get it right. So,
Sklarzik.
okay, Sklarzik.
Yes.
Sklarzik.
If
Okay.
we were back in the old country, it'd be Sklarczyk,
Okay.
but it's been pronounced Sklarzik.
Sklarzik. Okay. Well, no promises, but I'll try to get it right.
That
You
is
know,
not a problem.
I have a Y in my last name, and that freaks people out. I know. So.
Yeah.
And then you have a Z as well. So.
Yes, I've often said if you were to ever play hangman, it would be a good word to use because of all the consonants and the lack of vowels.
Sklarzik.
Excellent.
Okay. We'll see if I get it that way when I introduce you. I'll try. Thank you very much. Yeah, I went through. Yep. Yep. And there'll be an open bottle of water for you.
Are similar ones anyway. But it is really empty though, that's the worst.
Yeah, I'm sorry about that. Somebody has to go after break, so. I think we're going to go ahead and get started again, so if we could get folks to take their seats. Even though you don't think you'll need to keep track of time, I'll
No,
hit the start button and you can see it. So it's time to get started again. So our next speaker, moving along in the schedule, is Susanna Cooper. And Susanna is the Nematology and Entomology lead for the soybean breeding programs at Monsanto. And the title of her presentation is Breeding for Nematode Resistance, New Phenotypic Tools for a New Era. And we've got it started, and there you go.
I'm a little vertically challenged, so I don't know if there's a, yeah.
We'll lower that, and maybe
That'll.
if you could stand to the side without the risk of falling off.
Okay, sounds good.
Sorry.
One of the challenges of being short. So thanks you guys for the opportunity to chat today. I'm with Monsanto, and actually I went to Michigan State for my master's in entomology, so kind of good to see that. Yeah, I'm going to talk a little bit about what our team does, and so thanks for the invitation. So I do lead the Nematology entomology team for soybean breeding, so I was going to chat really briefly about what our team does, because when folks think about Monsanto, they think about transgenic traits, but there's a big effort around breeding for, move over a little, this?
Yeah, we'll turn it this way for you.
Okay. There's a big effort around breeding for native traits, and that's really kind of what my team does. So I wanted to kind of orient you a little bit about what my team does, and then talk about some of the efforts that the team is thinking through as far as improving phenotyping. So folks that work on nematodes know it's a really difficult thing to work on, and there's a lot of manual labor associated with it, so there's a lot of opportunities for improvement. And then really kind of thinking about, I can be like Donahue here. I'm thinking about where we'd like to be in the next few years. So what keeps the light on in our labs is really is pipeline characterization. And what that means is that if you open up an ASGRO seed guide and you look inside and you see the data around whether that variety is resistant or susceptible, that's coming from our team, especially in the context of nematode and insect pests. So my team does pipeline characterization for soybean cyst nematode, of course, root knot nematode, reniform, and then also insect pests such as soybean aphid and stink bug. And so that's a big piece of what our team does, and so we think about phenotyping all the time and how to do that better. So every week we're planting plants, we're taking down tests, and it's a big piece of what we're doing. And so we process a lot of samples every week and every year. So we think about phenotyping a lot within the team and how to make improvements on that. The other thing that the team also thinks about is also emerging pests, and really try to think about it in this context, because we do have limited funding resources, and there's only so much money for us to do our research. So a new nematode that we're beginning to work on is Melodia enterlobii, and that's only in a few counties in North Carolina, but it's potentially has a large presence in Brazil. And so thinking through that pest in this format, our first question when we're thinking about whether or not to work with a new pest is, how prevalent is it? So does it make business sense for us to kind of start thinking about developing techniques or working on that pest? Is there any chemical or biotech options available? So is there an easy solution that we can provide the grower to manage that pest? And if there is, then it may not make sense for breeding to approach that, because breeding can't turn on a dime. It takes us, if there's something floating in our germplasm, it might take us 5 years to increase the frequency in the germplasm, or it may take 10 years if we have to go back to wild PIs. The other piece, kind of thinking in the context that breeding can't move very quickly, is, can that pest adapt really quickly? So if we can provide a native trait, but it's going to adapt to that native trait in 1 year, it may not make sense for us to kind of think about working on that pest. Then how fast or slow growing is that pest? So do we think it's just going to be a regional problem, or is it going to be a problem that most soybean growers are going to be experiencing? So that'll help us kind of prioritize what we can work on. And then, so if it makes those prioritizations and we think it makes sense for us to work on it, the first kind of swath we do is basically check our pipeline material and see if we have anything that we can provide a solution for a grower immediately. So for instance, for enterlobii, we have some floating resistance for other root knot species, so the first kind of swath is can we find any floating enterlobii resistance in our pipeline that we can provide a quick solution to the grower. And the other big piece is, at the end of the day, our bosses are the stockholders, so we want to make sure that we can provide that our research dollars make sense for that. So a lot of times in the context of native traits, we're really not thinking about charging more for the seed, but maybe increasing our market share. So can we provide additional value so a grower will choose our seed over our competitors? So the other big piece is, if it does fit into that kind of that significance as being high, is the technical feasibility. So we're really fortunate with a lot of the nematode pests that there's a lot of techniques that have been developed in the academic sector that we can use and maybe increase the efficiency increase the efficiency within our team. But for some of the insect pests, this is not the case. So stink bug is one of the bugs we work on, and it's pretty difficult to screen for it out of the field. So whether or not it's going to be something easy to work on or not is another kind of piece that we kind of think about before working on a pest. So like I said earlier, really the big piece, 80% of what we do is really thinking about phenotype and product characterization, finding new QTL or resistance loci to provide new tools for the breeder. And so because we do that phenotyping quite a bit, that's kind of the big piece is what we're thinking about is how can we do what we do faster, quicker, cheaper. And these are the 4 buckets as far as when we're thinking about improving some of our processes. Greenhouse evaluation, that's going to be the lion's share of what the team does. But over the last probably few years, we've actually been tasked to help growers make better management decisions. And so what that means for us is helping them characterize their locations better so they can grow the right variety on the right acre of field. So there's a lot of really cool precision planting equipment out there that a grower has the option to potentially, on this section of the field, switch out a Peking resistant variety for an 8878 resistant variety. And so our team has been tasked with how do we make those recommendations for that grower. And so the last 3 buckets here is soil sampling, nematode enumeration from the soil, pathotyping, nematode populations. Those 3 kind of go into that kind of bucket of thinking about field recommendations for a grower and not around product characterization. So this is kind of what we do on a daily basis, and this is, like I said, this is a big piece of what the team does. And the sad fact about it is we've been probably doing very similar assay for decades, and it really hasn't changed too much. We're still germinating seeds, putting them in sand pots. We might have some nicer pipetters, but we're basically still putting a certain number of eggs on each plant every week. We let it go. The life cycle for SCN, it's 4 weeks. RKN, it might be a little longer. And then we basically clean up the roots, capture the cysts, and count those. And we're still using a dissecting scope. And it really, we might have a little fancier dissecting scope. We might be able to multiplex the root extraction, but we haven't made tons of we're doing very similar work that we did decades ago. One of the big focus areas for our team is around automating that enumeration component. And for folks in the room, I know we're not the only ones that are actually thinking through kind of automating this enumeration process. It's a big piece of what the team does. We have a suite of probably 4 people counting cysts half a work week, so it's a big. It's painstaking. It's an ergonomic stress. It's a big piece of what the team does. And so if we can alleviate that stress, our folks can do something a little more exciting than just counting cysts under a microscope. And we're not the only ones kind of looking at this type of technology. A lot of folks have been looking at trying to count cysts. This is a patent from Pioneer where they're utilizing, so we're really fortunate because soybean cysts have a unique shape and they actually autofluoresce. So this is a patent from Pioneer that they actually utilize the fact that the cysts actually autofluoresce. So they're shooting a wavelength at I believe around 470, and it's emitting at around 535, and so they're using that fluorescence to help them differentiate where the cysts are on the root and the root, and then counting those little dots. And this is another lab, Melissa Mitchum, who's in the room. She's kind of basically using a very similar, the autofluorescence component of the cyst, using a turnkey solution where you can actually have an instrument that actually captures the fluorescence, and you're able to count the cysts either on or off the root based on that fluorescence. We're taking a little bit of a different approach. We're actually just using our digital camera to take pictures of our cyst. And basically, because it has a unique shape and unique color and unique size, we're able to differentiate based on that category. So we're just basically using the attributes of the cyst to differentiate it from our debris. And this is my one data slide, so I'm going to spend a little bit of time on it. The one thing I got approval to share with you guys. So on the bottom here is the total number of cysts. And really what that means is that an individual took our counting dishes, put it under a dissecting scope, and counted every single cyst in that dissecting scope. We know it's not perfect, there's human error associated with that, but that's probably our gold standard as far as counting those cysts. What these blue dots mean is basically kind of what our current strategy for basically getting through all those plants in a work week. So we have a lot of plants, we have a finite amount of time and resources, and so what our team does for a lot of the material that goes through our pipeline characterization is they only count a subset of that dish and then multiply by what that subset is. So they basically subsample all the cysts that are extracted from the root ball, and then we multiply that. And so that's what basically this blue data is indicating is that potentially we counted 60 cysts, but our multiplication factor would have projected it being around 140. That seems a little bad, but we have really great resistance for soybean cyst nematode, and so really for our classifications as susceptible, moderately resistant, this estimation method really still works quite well for us. And then we have replications, and so we know there's some noise with our data, but it's still a great technique for what we're trying to do. Our algorithm is actually, what the algorithm, the red line is showing you is basically we've taken that same exact dish, we've counted it with a human eye, and then we put it in our imaging platform. And that's the data that we got back. And so when I talked with the electrical engineer to help us design this algorithm, really the instructions I gave her was if we can be in this same range, we're happy, but she far exceeded kind of what we were hoping. So the slope is near 1, which is awesome. That means that we're counting almost everything on the plate. The dots are really close to that line, so it's super, super accurate. So that's really exciting, and we're really excited about that, but that doesn't necessarily mean that we shift to that platform. What the team is really working through right now is how to further automate it. So it is laborious, it's a pain in the butt to count those counting dishes every time, but an individual can do it pretty quickly. They can take that counting dish, they can count it in about 30 seconds and go on to the next plate. When we think about putting it onto the imaging platform, if we actually have a human putting it on there, pressing take the picture, do it again, it's going to take more than 30 seconds. And so we're really working through the engineering around basically making that whole system automatic so that there is not a lot of human involvement as far as loading and offloading that sample. So, and that's always a cost of does it make sense for the assay. So for soybean cyst nematode, it's our most important assay, so it does make sense, but for some of the other minor pests, that tooling up cost might not make sense. So that's kind of the process that the team is working through right now, is kind of thinking through that tooling up piece. So the other big buckets that I'm going to talk with you about is really about kind of that field characterization and improving a lot of the techniques that we're thinking through on that. And it's a really exciting time for field work and a lot of stuff that we're doing, because there's a lot of off-the-shelf stuff that we can use to make our jobs easier. And really that's kind of what we're looking at. We don't want to, if there's something that we can buy that's off the shelf, we don't need to invent it. So, which is cheaper, and we can actually start using that stuff presently. So, and we're not the only folks that are thinking about doing soil sampling better, thinking about sampling zones. There's been work from a lot of folks in the academic sector doing this a lot in the context of cotton and root knot nematode, and also some in soybean cyst nematode. So we're not the first folks kind of thinking through these questions by any stretch of the imagination. But this is a publication, I don't know, a few years back, but it's pretty neat. It's in the context of soybean cyst nematode, and they're basically trying to kind of identify, be able to go back and identify zones that have SEN and predictable zones. And so they have this, I think it's around a 5-hectare sampling zone. They have kind of a neat sampling strategy where they're doing kind of a fractal nested sampling, and they're really just trying to see if they can predict areas of pressure year to year. And they were able to, and I guess that's not super surprising, but when we're thinking about this and providing this as a recommendation for a grower, the way they did it is not feasible as far as the number of samples and going out and hand sampling. So, you know, a lot of the growers we're working with have hundreds of acres, you know, so you can't have an army of folks going out and soil sampling and providing these recommendations. When we're thinking about providing recommendations on various areas of HG types or just nematode numbers. And so, like I mentioned earlier, there's a lot of really cool stuff that we have at our fingertips. A lot of automated soil sampling that you can imagine. You can put some auto steer on there and really just have it go through the field and basically know where have it geotagged and know exactly where you're sampling and develop your nematode maps that way. So this is a neat soil probe that's you can mount to a UTV or ATV, so you don't have a lot of soil compaction, which is pretty neat. And it still takes some human interface with it for sure. You're unloading the core and you're putting it into your bag. It's pretty fast, but the real watch out here is that if any of your questions are in season, you can't go through rows or overplant. So it's something that you would probably just use before or after your crop is in the field. Here's another one. It's kind of a big machine, and I think it's around $50,000. It's pretty expensive, but the neat thing here, and it's not very high throughput, but the really kind of neat thing to think about with this one is that this hose here basically sucks up your soil. It goes into a conveyor belt, and it automatically puts that soil into your bag for you and geotags it. So it may not make sense in the context of nematodes, but I mean, they're really thinking through some cool engineering issues as far as sample handling and reducing that human interface. This one's pretty neat as well. This is on a rover. So this is something that you can do if your questions require you to actually go into your soybean field. You can actually take soil samples in season. The watch out here is it's an auger, and at least from some limited work that we've done, you know, augers, and most of the folks in the room probably already know that, but probably you want a core over an auger. But it is kind of interesting to think about, you know, some technologies that will allow you to do some in-season monitoring. And this is one that is pretty neat. It's a totally different way to take a soil sample. It's actually more like a potato shoe, and it basically takes your soil and automatically puts it into your collection vial, it's barcoded, and it geotags it for you. So just some different, I think there's one more. This is another one that it's basically mounted behind a tractor. It goes through your field really quickly. If you ever go and see a YouTube video, it's pretty neat. The one watch out here is for nematodes. Again, it may not make sense because you don't have a lot of, the depth of the soil probe is finite, so I think it's around 6 inches, and it may not make sense for questions regarding nematodes, but it can go through the field and composite soil pretty quickly. So, I mean, there's a lot of stuff that you can imagine putting some auto steer on or some GPS stuff on there and really kind of help us do our jobs easier and quicker. And not really require that army of folks to get to those answers. The other big piece that the team has been really working on and thinking through how to basically, so now we can hopefully get those soil samples to our lab easier, not as much manual labor. We can maybe do higher density studies to kind of answer some just fundamental questions about SEN distribution, SEN yield loss, but we still have to process those when they come back to our lab. And you know, the current gold standard method is really doing soil elutriation, which is a great technique, but it's pretty dirty, and it takes someone pretty skilled to run the machine. So you have your 100 cc of soil, you put it into this funnel, it floats out the cysts, and then you capture the cysts and you crush them and you count the eggs, and you can provide a recommendation to the grower based on the cysts that were in that soil. There's a lot of neat techniques. There's a lab in Australia that's been doing a lot of qPCR-based soil sampling and making providing for other pathogens than the nematodes. So there's a lot of primers in the public literature to help you think through kind of, qPCR-based analysis, and some study that's done some really good correlations with CT levels and egg counts. And so the big watch out here is really kind of that we're trying to work through is really thinking about that soy subsampling component, because right now when we are putting into the elutrator, we're using 100 cc, and the cost of the kits and the DNA extraction is quite expensive. So we have to kind of think through the best way to subsample that initial sample. But our team can process, I don't know how many we can do. This can probably do 200 or 300-fold what our team can do with the manual method, or more so. So it's really transforming what we can ask. And so, I mean, that's the really exciting thing for the team is that we have these tools that we could get crude answers to before, but hopefully we can develop some more elegant experiments that we can really kind of answer some really just fundamental questions that have been hard to answer with older methodologies. And so right now the team is really kind of thinking through, better, like I mentioned earlier, better ways to do our pipeline characterization, and then also better site characterization to provide growers recommendations on what varieties to plant where. And really that's all around species level. And really the next step is around basically thinking about that pathotyping or HG typing or race typing, whatever kind of system you're thinking about for in the context of SCN. And as folks who work with SCN know, this is not it's not a quick process. So how it works presently is we get our soil samples, we extract out all the nematodes and the nematode eggs, and there's not enough, so you can't just go from there and put it on your panel of differentials. We have to put it on a susceptible plant, grow it a number of generations, so 2 to 3 generations in the greenhouse. Now we have enough worms that we can actually put it onto our panel of checks and see what checks it can or cannot grow on, and then we can provide that recommendation to the grower. So when the grower is thinking about buying seed, he's doing that in the winter months. And so if he gives us a fall soil sample and we don't give him data back till April, it's not going to change his management decisions. And so we have to find a way to do this faster so he can actually change his buying choices. And so there's a lot of strategies that folks are thinking about, with sequencing being so inexpensive now, trying to find molecular markers to help us identify that virulence classification is really where we're hoping to be. And with qPCR, you can think about that, potentially the question isn't yes or no, but you can say frequency. So we're imagining that we get that same soil sample, we come to our team, and we could run qPCR and say 20% of the population is virulent to 8878, 5% is virulent to Peking, and that can help us help the growers make those management decisions. And hopefully we can say in this area of the field, there's no nematodes, so you may not need any resistance in that area of the field. The other section of the field is a high population, and it's virulent to 8878, so you might need a Peking variety. And so that's really where we're hoping to be in the next, in the future. And really, this just the fundamental science for us to enable us to get those tools is hard, but we're making great strides in getting there. So, gosh, that was really, really quick. And that's really all I have. I have a lot of time for questions, and folks at Monsanto, we don't work in a vacuum. There's a lot of folks that help us with all these projects, and so there's a lot of folks that helped us kind of think through these questions and kind of do what we do better. So with that, I'd be happy to take any questions.
Questions?
Sure.
How active is Monsanto grading for resistant sources beyond 8878?
Yeah, how active? It's an increased focus for sure, and really So a big focus for Monsanto is around soil, and we actually have a pretty expansive survey that we've been conducting over the last 3 years within our breeding locations primarily, and it's really what's the word I'm looking for? It's really demonstrated to the breeders that they really need to start thinking about alternative resistance to 88788. And so there's folks within my team that are trying to develop better sources, so the breeders will actually have that be more of an active part of their program. But breeding can't turn on a dime, so there's a handful of Peking variety. Every year there's a handful of Peking varieties. They don't necessarily get chosen to be commercially launched, and there's a couple of Hartwig. There's a Hartwig variety, I believe, that was on the market for a couple of years, and growers didn't purchase it, so it fell off the market. But we have active breeding programs in Peking and Hartwig. Hartwig would primarily be in probably mature group 4 and later, and Peking probably throughout the maturity groups. But it's increased effort, and hopefully in the near future you'll see more product offerings. I know that's a big watch out, right? When you're thinking about, so it's something that we think about, because if we provide, there's not a lot of options for the growers. Even if you're going to Asgrow or Pioneer, there's just a handful of non-8878 varieties out there, and yield is also an issue as well. So, but it definitely increased effort, especially with the soil surveys, really kind of really increased the effort. So it's easy to convince people when you have that data showing it. But the other big piece I would like to also mention about the fact of when you're thinking about HG typing or race typing, the other thing that we're thinking about is that 10% criteria, right? So we still also think there's a lot of value so you can have something that's categorized as virulent to 8878, so that's just 10% of Lee, right? So, or 10% on there. And so we're seeing with some of our trials that 8878 still performs very well in fields that potentially have been characterized as being virulent to 88788. So there's still a lot of value, and so we're really trying to elucidate that too. Is like where is that breaking point where we need to have that peaking option too? Because it may not be 10%, it might be something higher. So, but still trying to think through that. But hopefully you talk to us in 2 or 3 years, and I can say there's more products in the pipeline. So, yeah.
Other questions? I guess.
Root knot. So I would say anything maturity group 5 and later has root knot resistance in it. There's probably nothing in our product, very minimal stuff that has. So it's a must-have for 5 and above. So everything has incognito resistance. There might be a handful of things in our pipeline that isn't, but it's almost for the commercial team, it's a must-have for 5. And the maturity group 4, I'd say it's probably 20 to 30% of the pipeline. So yeah, in North Carolina, you can't grow a soybean variety without root knot. So, and a lot of those, so it's quite critical. And then in Brazil, even more critical. So, right, so that's a big effort. I didn't even mention that here because a lot of, SCN is number 1, but root knot is very important in the South and also in Brazil. So, yeah, a lot of our product portfolio does have root knot in it. Yeah. It's a requirement. So.
Excellent. Thank you very much, Susanna.
No problem.
Perfect. As we transition to the last but not least presenter of the day, or the morning, it's my pleasure to introduce Dawn Sklarczyk, and Dawn comes to us highly recommended from a colleague of ours, George Bird, so I want to thank him publicly as well for hooking us up with Dawn. And Dawn is owner and operator of Sklarczyk Seed Farms in northern Michigan, and he's a state and national advocate for research resources for scientists, and has really integrated science into real-world crop production. And in fact, the title of his presentation is Advances in Science to Meet Real-World Crop Production Needs. And so we're going to turn it over to Dawn.
Thank you, and I appreciate the introduction, especially the enunciation of my last name, which is very challenging for anyone. But I'm very humbled to address this group, and hopefully I can give you some thoughts to think about from a different perspective, the grower perspective. Now, the value of this photograph is that it's actually of my father's great uncle, and he is tilling some ground that references a sturgeon river. As it turns out, the Sturgeon River is some 20 miles from our farm operation, but our farm is the southernmost headwaters for that river that feeds into the Great Lakes, that is one of the tributaries that is considered very pristine for the ecology of the area itself. Now, I'll start off with a question. Can human health issues assist in food crops? In the early 1980s, previously healthy gay men in Los Angeles developed a rare lung disease. Later, it would be called AIDS. The ELISA test was developed as a diagnostic tool to help detect AIDS. That research helped with the advancement of the ELISA test for plants and plant pathogens as well. Now, could ELISA testing change crop production practices? My father's generation relied upon visual observation to determine the health of their crops. They used a system of tuber-to-tuber reproduction, a process that reproduced latent bacteria and symptomless viruses. What they would do is take individual family groups, harvest them in the fall, place them in a bag. They were numbered, sent to a warm climate. An inspector would then go through and analyze each of the plants. If there was one that did have a disease, that family was destroyed. The remaining number of good families were planted, and that was the start of the seed program. The system relied only on visual observation for plant health, and it required many years of increasing in the field to provide the amount of seed necessary to make a crop that was of value. The floriculture industry was already using tissue culture for rapid multiplication, but tissue culturing of potato plants would be worthless unless mother plants were known to be as free as possible of bacteria and virus. With the development of the ELISA test for potatoes, new opportunities were now available for rapid expansion of specific clean genetics. But first, the potato industry had to realize that it held value. The concept of producing potatoes the seed potato farmers had used for years was going to be challenged. And change was not an easy thing for the potato industry to embrace. Yeah, I think that's the same for other crops today. The change from a tuber-to-tuber system required support from the science community. Several researchers recognized how the use of tissue culture could benefit the potato industry and advocated for its adaptation. Sklarczyk Seed Farm was the first farm east of the Rockies to produce tubers in a tissue culture laboratory in the protection of a greenhouse-grown environment for an on-farm production capacity. The first crop that we produced very few tubers. Now, what were the benefits of the new concept for seed potato production? 100% of the plants used would be tested with the use of ELISA and other testing methods to verify the freedom of symptomless bacteria and latent viruses. The tissue culture laboratory allowed for rapid expansion of selected family groups, so thousands of clone plants could be produced in a few months. Each month, the mother plant could be increased by a 5x. The plants would then be placed in the protection of a greenhouse. Then follow-up testing would verify that there was not any infection while in the greenhouse. Serving as the chairman of the Michigan Potato Industry Commission, it provided contacts for me at Michigan State University. I soon recognized the importance of communicating with key people. Researchers at Michigan State University connected me with researchers in other states. The conversations were invaluable to provide answers to my many questions. While attending a plant microbiome and insect genomic resource improvement workshop, I had other contacts that turned out to be very valuable. I quickly realized that researchers had a passion for their work and were glad to share it with production agriculture. At the same time, I became more involved with the National Potato Council, serving on many of their committees, serving as a vice president and ultimately the president of the National Potato Council. With tissue culturing of potatoes, rapid reproduction of a specific clone was now possible. The previous practice would require a lifetime of field increases to match the ability to reproduce a specific genetic line in 1 year. An example would be if you started with 10 plants, in 6 months you could have 31,000 plants, enough to fill a large greenhouse. All the tubers would be free of any tested diseases or pathogens. The healthy tubers were able to withstand adversities, much like when we are not fighting off a cold, we feel much better and can go about our daily activity with a better performance. The seed potato industry's response was not that receptive. Seed growers didn't want change. The orders for the tissue culture seed potatoes, our mini tubers, from seed growers were slow to come, and what came in small volumes. I had to come up with a way for the potato industry to recognize the value of tissue culture for the potato industry. It required going around the seed grower, providing seed directly to the commercial grower so they could plant trials. They saw the benefits. They requested their seed suppliers to use tissue culture-derived seed. Now, at the same time, commercial growers were experiencing yield declines, which drove part of their desire to look for what was taking place. They were asking questions. What can we do? What is taking place? Now, I knew seed was not the only answer, but it was an answer that I could work on. What developed was growing with pasteurized soil mix in a greenhouse environment. Now, all the equipment that I use had to be custom made. What I found early on is plant mortality was extreme. When we transferred the little plants from the lab to the greenhouse, we had a difficult time having the plants survive. I had many learning experiences in the production process. While this was happening, each year more and more requests for mini tubers were being received, but the production by each plant was averaging only 1.5 tubers per plant. Some of them were very large. We attempted to come up with different ideas to control the size of the seed potatoes. Growing in wooden beds as we were was not the answer, but what it did for us was to recognize that the potatoes enjoyed producing tubers along the edges of the boxes. So our first attempt was to create additional divisions within the box to create more tubers. At the same time, we were working with a researcher or a graduate student at Michigan State University trying to come up with new ideas to allow us to have greater production. That required this researcher to drive over 3 hours to do his research work at Michigan State, which really was taking place at Sklarzik Seed Farm. His research was successful. It resulted in greater root development, so the number of tubers increased. He later based his doctoral thesis upon that work, and now many other crops enjoy the benefits of his work that he performed. Now, the process that we were using, that of being artificial growing medium, had drawbacks. So as I mentioned, we tried divisions into boxes. Then we tried individual pots for the plants. We then took the step of sequential harvesting the pots several times during the year, pulling out the tubers that could be reached from the outer edges. But we still lacked control of the tubers. Large tubers were then cut, which created an open wound for soil pathogens to invade the tubers. They were also difficult to plant, especially with mechanical planters. We realized that planting in pots was not the answer. So we had a conundrum. What were we going to do? Serving on an AAR Agricultural Research Service Review Committee turned out to be very beneficial for me. I was one of 4 to review projects related to the potato industry. It allowed me to have a voice in the ARS budget. I also was invited to serve as one of the reviewers for the potato plant breeding projects, which was a budget of $3.5 million. I was able to connect with researchers from other areas and also connect researchers from those areas with MSU researchers. It made me realize even more the importance of collaboration across state lines about the researchers doing work not only within their own disciplines, but other disciplines as well. At the same time, the importance of soil health for the potato industry was starting to be noticed. And the question was, what factors are causing the yield declines in the fields? At that point in time, I was the president of the National Potato Council and took trips into Washington, D.C. to justify the return on the value for the research dollars. It was easy for me to show Congress that it was justifiable. One thing that sticks in my mind is while standing and waiting to get on an elevator, one of the congressional staff members came over and was going to get into their selective elevator, and he said, "Don, what are you doing in Congress?" "Well, I know you're talking about potatoes." I said, "Yes, specifically research dollars." He said, "Go into my office and ask my chief of staff to have a conversation with you," because I explained to him how the research that was done in 1 year was provided to the growers for the next year's production. He ended up by saying, "Go into my office and tell my chief of staff he needs to listen to this story. We're going to be discussing budget funding this afternoon." I found that very rewarding and self-satisfying that it made my trip into Washington, D.C. at that time very well worth it. Now, getting back to our own operation, I recognized that we had to do something different. But what justified the change to a new system? The system would be a nutrient film technology that we were going to have to develop. I had questions. I wondered if, while testing for the major pathogens, could untested infections be still taking place for the tubers we were delivering? Now, the demand for the mini tubers continued to increase to a point that our operation was at its capacity for production. No attempt that I was using to control the size would work. I wanted to deliver mini tubers that could be planted as whole tubers and realized that cutting of seed tubers provided an opportunity for soil pathogens to invade the tubers. So my response was to convert 2/3 of our operation the very first year. My previous success made me forget about basic science and what I should have been doing. It was a disaster. Other crops were produced in NFT, but they were all harvested above their coverings. What I had to do was come up with a method so that tubers could be harvested a number of times while the plants continued to grow and produce more tubers, but still not destroy the plant. The ideal tuber size for planting in the field was yet to be determined. There was no previous work that I could draw from potatoes. Even the covering type that would be used was unknown. This first attempt by using black plastic, which I thought would enhance their growth, turned out to be the worst color that I could have had because of the solar radiation that came off the plastic. Pretty much took away about 1/3 of our plants the first year. The style of the tray, the distance between the plants, everything had to be determined. We made many other discoveries. The importance of starting with nothing but H2O was quickly learned. We had to set up a reverse osmosis fluid system and then realized that each variety had its own recipe that needed to be formulated. Otherwise, the plants responded differently. Even during their growing cycle, we had to change what we provided for their growth. Without an abundance of oxygen, we found that no matter what nutrient load we gave them, we were not getting the response that we expected. Now, plant survival was critical for success. We recognize that. Our goal was for 100% of the plants to produce tubers, and you can see in this slide that we had probably a 40% survival rate. Extensive records and graphing provided some base information. Several tray designs were looked at. Even the tray orientation for harvest efficiency was explored. The NFT process was full of challenges for us, but we were getting better at it all the time. But as we developed the ability to produce the tubers, we found that it was only the first of the challenge that we had. You see, the tubers lacked any skin set when they were harvested. Again, I had to turn to the research community. The first year that we produced the NFT tubers, by the time we were ready to ship them, they resembled more like raisins than they did tubers because of the dehydration that took place. The best storage practices at the time were not acceptable for the extremes that we were creating. At Sklarzik Seed Farm, we had to develop a post-harvest and a long-term storage. Our facility became a research center for a major supplier of potato and vegetable storages. I learned quickly that producing a crop was only the first step in providing the desired quality that the industry required. Now, the research that we did that was taking place resulted in new standards for the industry today. Now, what were the benefits of having whole seed? Uncut seed prevented the opportunity for soil invasion or for the invasion of soil pathogens as the plant develops. Whole seed was much easier to plant. Seed of a specific requested size was now possible by our operation because our staff would literally go through twice a week and harvest the ideal size tuber. If a tuber was 14 millimeters on Monday, by Thursday it would be up to 18 millimeters. The industry's accepted size was between 15 and 17 millimeters. Even with all that was taking place in our ability to increase our production, it still cost over $20,000 an acre for just the seed to plant an acre of base seed potatoes. What was taking place is the tubers were expanding with little skin. Fortunately, there was a researcher from North Dakota that had dedicated his career to skin set of potatoes. And he and I had many hours of conversation about what was happening. The dehydration of the tubers was explained to me, so what I had to do was come up with a way to create an artificial skin after harvest of the tuber. So a greening process had to be developed. I again turned to research, but this time it was reverse research, because what I found was I was looking to what caused greening in potatoes when they were marketed in a store. And the research told me the light spectrum that was the most disadvantages, or that created the most disadvantage for stores to have in keeping potatoes ungreen. So I gravitated to that spectrum and set up a process. So a new storage was constructed, found that if we stored our harvested potatoes for 2 weeks at 55 degrees Fahrenheit, 97% relative humidity without free moisture, with less than 0.2 of a degree temperature variation, we could create a artificial skin, or a skin on the potato. We had to do that because some of our first harvested tubers has to be stored for over 14 months. And unlike potatoes that you receive in the store, we could not have any artificial sprout suppressants on the potatoes themselves. Now, the NFT process eliminated any possibilities of pathogens transferring from the soil to the growing medium. What happened over time was the NFT process was accepted around the world. At this point in time, Sklarzik Seed Farm is shipping potatoes to Mexico, Chile, Thailand, Vietnam, Egypt, Brazil. We, Sklarzik Seed Farm, our mini tubers are the first ever to be allowed to be shipped into Chile and Mexico without any restrictions. It took collaboration with a number of agencies within the United States and Chile, and reconnaissance missions from the governments to allow this to accomplish, but it does prevent any pathogens, any soil pathogens, from being a possibility within our process of production. In 2015, our production will be over 6 million mini tubers. That 1.5 tubers per plant that we had initially is now based at 20 tubers. With the use of artificial lights, we can have 40 tubers per plant. Now, to get that in perspective, every 20 mini tubers that we produce will result in a truckload of potatoes for the Indian market. So that means our production next year will result in over 300,000 truckloads of potatoes. So the question would be, what do we have to do to get that same type of turnaround for our soil issue? Now, how did we get from 1.5 tubers per plant? We installed in-floor heat. We put hydro-cooling pads, variable frequency controls on the fans and the pumps. We've got solar collectors, waste oil burners. Computers monitor everything. All the environmental readings are 2-minute increments. So every storage aspect and growing aspect is recorded. But most importantly, connecting with the research committee or the community allowed us to have advances in science to help our production needs. Now, the sources for research funding are being reduced each year. Any researcher realizes that. At the same time, the cost of research that you're doing becomes more complex. So what must we do? Can partnerships develop with other research communities? I look at what happened last year where scientists from around the world had to do these genomic sequence of potatoes. It started with a new complex way of solving a problem. Can other crops do the same thing? Can soil plant health and human research benefit each other? Now all crops are looking at soil health, and I'll call it the good bugs that are there. So here's a wild dream. Can a formation of a group with as much passion as the Humane Society be formed to advocate for research funding to raise funds for the research community? So new ideas will never cease. Something that we started working on last year is a project that is producing cherry tomatoes on the top while producing potatoes in the lower portion. It's a grafting process. A company from Europe is the base company. They call it ketchup and fries. The original order was for 10,000 plants. This year we produced 70,000. The order for next year is 1 million plants for 2016. So the value of the partnerships will keep on increasing. We would have never been able to accomplish what we did without connecting with the research community. You know, growers must tell the story to Congress to maintain the present sources of funding. And additional partnerships must develop beyond the present collaborators for funding sources. Who that will be with, we don't know at this time. The only thing that we know for sure is that researchers, they need to continue to do the great work that has been done, because the accomplishments and the growth at Sklarzik Seed Farm would have not been possible without the research community's assistance. So thank you to the research committee for all that has been done. And thank you for allowing me to present today.
Questions for Don? I guess one question I was sitting thinking is, and it might be difficult to answer off the top of your head, but what are some specific research needs that you have that are unmet? Is there any research topics? If you were in charge of $10 million of research funds for the potato industry, what general area or specific area do you see could use the most funding, or is the biggest research need?
At this point in time, the greatest research need that I can see would be related to soil health. I believe that is front and center to everything else that's taking place, and that can spin off to the type of development of new technologies to find out what's taking place in the soil, what's causing yield and quality declines. I think that is really driving what will happen to feed the next generation. And I don't know if everyone realizes, but I'm very passionate of potatoes. But potatoes is becoming a very well accepted worldwide crop of feeding the masses. So we need to use potatoes as a leader to try to come up with new ways of increasing the production or maintaining production of what we had before.
Excellent. Other questions for Don? Here's one.
Today, what is your concept of soil health, and how does a grower recognize that maybe his soil has become degraded? That is a very difficult question, because just as it was hard for the seed potato industry to recognize the need for change and develop and accept new production practices, I think growers are slow to recognize what's happened to their soil practices in the past. So how we do that is going to be incremental steps and doing research much like what is taking place, to show the growers firsthand that sometimes producing a crop in a rotation that they presently have is actually costing them more money than what it's saving. And to get the story to Congress, to people who control those budgets, I think is going to be invaluable for us.
Excellent. Thank you, Don. So we're fortunate to have all of our other speakers still here in the room as well. So we'll open it up to general questions of any particular kind for any of the speakers. Anybody? We have one here. Yes, Augie? And could you repeat the question because the others?
The question would relate to how do we transfer the information to the grower community, the research that is being done. Is that paraphrasing it? I believe agriculture is changing so much that it will rely more than just on extension. It's direct contact to the growers themselves, because the growers have their own researchers on board today. So reaching out to them, and it's a struggle. As a research chairman, I'm struggling with that right now. How do we, I hate to say force feed, but really that's what has to happen. How do we get that information? Doing work like what Dr. Bird is doing today, I believe, is the first step. And that's obvious that some of the other commodities besides potatoes are recognizing the need for it. So he's asked to participate in seminars, which is a great thing for us, but it adds a burden for him. So I think that's going to be it, trying to reach out and force feed that information to them, giving actual information to show realistically it's costing them money to do what they're doing today. Because if the pocketbook is impacted, it'll get their attention. And that's what happened with the transfer with the new technology for seed.
Thank you, Don. Other questions or comments? Yes, Gui Ping? So the question was that Susanna had. Okay. Other questions? Going once? Going twice? Well, if there are no other questions, I'd ask you to join me and let's thank all of our speakers again today. Thank you very much. Very good. think that's it.
Some seeds of thought.